Chapter 13 Photosynthesis in Higher Plants. 13.1 What do we know? 13.2 Early experiments. 13.3 Where does photosynthesis take place? 13.4 How many pigments are involved in photosynthesis? 13.5 What is light reaction? 13.6 The electron transport. 13.7 Where are the ATP and NADPH used? 13.8 The C4 pathway. 13.9 Photorespiration. 13.10 Factor affecting photosynthesis. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants during photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. 13.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, green pigment of the leaf, light, and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black pepper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaf in the presence of light. Another experiment you may have carried out where a part of a leaf is enclosed in a test tube containing some chaos-soaked cotton which absorbs CO2, while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? 13.2 Early experiments. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804. In 1770, performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plant restored to the air whatever breathing animal and burning candle remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think to light up the candle without disturbing the setup? Using similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight. Jan Ingenhousz, 1732-1799, showed that the sunlight is essential to the plant process that somehow purifies the air formed by burning candles or breathing animal. Ingenhousz is an elegant experiment with an aquatic plant showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plant that could release oxygen. It was not until 1854 that Julius von Zach provided evidence for production of glucose when plant grows. Glucose is usually stored as starch. His later studies show that the green substance in plants, chlorophyll as we know it now, is located in special bodies later called chloroplast within plant cell. He found that the green parts in plant is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiment is done by T.W. Engelmann 1843 to 1909. Using a prism, he split light into its spectral component and then illuminated a green alga, Cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the site of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B, as discussed in section 13.4. By the middle of the 19th century, the key features of plant photosynthesis were known, namely the plants could use light energy to make carbohydrate from CO2 and water. The empirical equation representing total process of photosynthesis for oxygen evolving organism was then understood as equation CO2 plus H2O in the presence of light gives CH2O plus oxygen, where CH2O represented a carbohydrate, for example glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by microbiologist Cornelius van Nil 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. This can be expressed by equation twice H2A plus CO2 in the presence of light gives twice A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to oxygen. Some organisms do not release oxygen during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not oxygen. Hence, he inferred that the oxygen evolved by the green plants come from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore equation 6CO2 plus 12H2O in the presence of light gives C6H12O6 plus C6H2O plus 6 oxygen, where C6H12O2 represents glucose. The oxygen release is from water. This was proved using radioisotope technique. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? 13.3. Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplast. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does take place in the green leaves of plants, but it does so also in other green parts of the plant. Can you name some other plants where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surface parallel to the wall? When will they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn form starch. The former set of reactions, since they are directly light driven, are called light reactions or photochemical reactions. The latter are not directly light driven but are dependent on the products of light reaction ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions or carbon reactions. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. 13.4. How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves even in the same plant? We can look for an answer to this question by trying to separate the leaf pigment of any green plant through paper chromatography. A chromatographic separation of leaf pigment shows that the color that we see in leaves not due to a single pigment but due to four pigments: chlorophyll A, bright or blue-green in the chromatogram; chlorophyll B, yellow-green; xanthophyll, yellow; carotenoids, yellow to yellow-orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelength. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb light of different wavelength. Figure 13.3. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the vigor. From figure 13.3a, can you determine the wavelength color of light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelength too? If yes, which one? Now look at figure 13.3b, showing the wavelength at which maximum photosynthesis occur in a plant. Can you see that the wavelength at which there is maximum absorption of chlorophyll A, i.e. in the blue and the red region, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that the most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does takes place at the other wavelength of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping the light, other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from oxidation, photooxidation. 13.5. What is light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. Several protein complexes are involved in the processes. The pigments are organized into two discrete photochemical light harvesting complex LHC within the photosystem 1 and photosystem 2, that is PS1 and PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The light harvesting complex are made up of hundreds of pigment molecules bound to protein. Each photosystem has all the pigment except one molecule of chlorophyll A, forming a light harvesting system called antennae. These pigments helps to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystem. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence called P700, while in PS2, it has absorption maxima at 680 nanometer, and is called P680. 13.6. The electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electron to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of cytochrome. This movement of electron is downhill in terms of an oxidation, reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigment of photosystem 1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy-rich NADP+. The addition of this electron reduces NADP+ to NADPH2. This whole scheme of transfer of electron starting from the PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electron, transfer to another acceptor, and finally downhill to NADP+, reducing it to NADPH plus H+, is called the Z scheme. Due to its characteristic shape, this shape is formed when all the carriers are placed in a sequence on a redox potential scale. 13.6.1 Splitting of water. You will then ask, how does PS2 supply electrons continuously? The electrons that were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with PS2. Water is split into twice H plus, oxygen and electrons. This creates oxygen, one of the net product of photosynthesis. The electron needed to replace those removed from photosystem 1 are provided by photosystem 2. Twice of H2O gives 4H plus oxygen plus 4 electron. We need to emphasize here that water splitting complex is associated with the PS2, which itself is a physical located on the inner side of the membrane of the thylakoid. Then, where are the protons and oxygen formed likely to be released, in the lumen or on the outer side of the membrane? 13.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bond. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS2 and then the PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening is in the stroma lamellae. While the membrane or lamellae of the grana have both PS1 and PS2, the stroma lamellae membrane lacks PS2 as well as NADP reductase enzyme. The excited electron does not pass on to NADP+, but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence result only in the synthesis of ATP, but not of NADPH plus H+. The cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation. 13.6.3 Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time, these are the membranes of the thylakoid. There is one difference though. Here the proton accumulation is toward the inside of the membrane, i.e. in the lumen. In respiration, proton accumulate in the intermembrane space of the mitochondria when electron move through the ETS, as you have read in chapter 14. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electron and their transport to determine the steps that cause a proton gradient to develop. A. Since splitting of the water molecule takes place on the inner side of the membrane, the proton or hydrogen that are produced by the splitting of water accumulate within the lumens of the thylakoid. B. As electrons move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located toward the outside of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes the proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane. Along with electrons that come from the acceptor of electron of PS1, protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One, called the CF0, is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer side of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. Energy is used to pump proton across a membrane to create a gradient or a high concentration of proton within the thylakoid lumen. ATP synthase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electron, the ATP will be used in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugar. 13.7. Where are the ATP and NADPH used? We learned that the products of light reaction are ATP, NADPH and oxygen. Of these, oxygen diffuses out of the chloroplast while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugar. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the product of the light reaction, i.e. ATP and NADPH, besides CO2 and H2O. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss amongst yourself. Let us now see How the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2N or sugars. It was of interest to scientists to find out how this reaction proceeded, or rather what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among several efforts to put radioisotopes to beneficial use, the work of Melvin Kelvin is exemplary. The use of radioactive carbon-14 by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it was called Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation, or whether any other products was formed in other plants. Experiment conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA, since then CO2 assimilation during photosynthesis was said to be of two main types. Those plants in which the first product of CO2 fixation is a C3 acid, phosphoglyceric acid, i.e. the C4 pathway, and those in which the first product was a C4 acid, oxaloacetic acid, i.e. the C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later. 13.7.1 The primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have, which after accepting fixing CO2 would have 3 carbons of PGA? These studies unexpectedly showed that the acceptor molecule was a 5-carbon ketose sugar, ribulose bisphosphate, RUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C4 acid, the primary acceptor would be a 2-carbon compound. They spent many years trying to identify 2-carbon compound before they discovered the 5-carbon RUBP. 13.7.2 The Calvin cycle. Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP is regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outset understanding very clearly that the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathway. For ease of understanding, the Calvin cycle can be described under 3 stages: carboxylation, reduction, regeneration. First, carboxylation. Carboxylation is the fixation of CO2 into a stable intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme ribulose bisphosphate carboxylase, which result in the formation of 2 molecules of 3-phosphoglyceric acid. Since this enzyme has also an oxygenation activity, it will be more correct to call it ribulose bisphosphate carboxylase oxygenase, or RUBISCO. 2. Reduction. These are a series of reactions that lead to the formation of glucose. The step involves utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of 1 molecule of glucose from the pathway. Third, regeneration. Regeneration of the CO2 acceptor molecule ribulose bisphosphate is crucial if the cycle is to continue uninterrupted. The regeneration step requires 1 ATP for phosphorylation to form ribulose bisphosphate. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycles are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Calvin pathway. It might help you to understand all of this if we look at what goes in and what comes out of the Calvin cycle. See table. In and out. In 6 CO2, out 1 glucose. In 18 ATP, out 18 ADP. In 12 NADPH, out 12 NADP. 13.8. The C4 pathway. Plants that are adapted to dry tropical region have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then, in what way are they different from C3 plant? This is a question that you may reasonably ask. C4 plants are special. They have a special type of limb anatomy. They tolerate high temperature. They show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical section of leaves, one of a C3 plant and the other of a C4 plant. Do you notice the differences? Do both have the same types of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 plants are called bundle sheath cells, and the leaves which have such anatomy are said to have Kranz anatomy. Kranz means freth and is a reflection of the arrangement of cell. The bundle sheath cells may form several layers around the vascular bundle. They are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of the leaves of C4 plant, maize or sorghum, to observe the Kranz anatomy of the distribution of mesophil cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical section of the leaf. Observe under the microscope. Look for the bundle sheath around the vascular bundle. The presence of the bundle sheath will help you identify the C4 plant. Now study the pathway shown in figure 13.9. This pathway that has been named the Hatch and Slack pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate (PEP) and is present in the mesophil cells. The enzyme responsible for this fixation is PEP carboxylase or PEPase. It is important to register that the mesophil cells lack Rubisco enzyme. The C4 acid, oxaloacetic acid, is formed in the mesophil cells. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophil cells itself, which are transported to bundle sheath cells. In the bundle sheath cell, these C4 acids are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophil cell where it is converted to phosphoenol pyruvate again, thus completing the cycle. The CO2 released in the bundle sheath cells enter the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme ribulose bisphosphate carboxylase oxygenase (Rubisco), but lack PEPase, phosphoenol pyruvate carboxylase. Thus, the basic pathway that results in the formation of the sugar, the Calvin pathway, is common to the C3 and C4 plants. Did you note that the Calvin pathway occurs in all the mesophil cells of the C3 plants? In the C4 plant, it does not take place in the mesophil cells but does so only in the bundle sheath cells. 13.9. Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plant: photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Calvin pathway, the first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form 2 molecules of 3-phosphoglyceric acid, that is catalyzed by ribulose bisphosphate carboxyl oxygenase, I Rubisco. Equation: RuBP plus CO2 in the presence of Rubisco gives 2 times of 3 phosphoglyceric acids. Rubisco, that is the most abundant enzyme in the world, do you wonder why? Is characterized by the fact that its active site can bind to both CO2 and oxygen, hence the name. Can you think how this could be possible? Rubisco has a much greater affinity for CO2 when the CO2 is to oxygen is nearly equal. Imagine that would happen. Imagine what would happen if there were not so. This binding is competitive. It is the relative concentration of oxygen and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some oxygen does bind to Rubisco and hence CO2 fixation rate decreases. Here the RuBP, instead of being converted to two molecules of PGA, binds with oxygen to form one molecule of phosphoglycerate and phosphoglycolate, 2 carbon, is a pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugar nor of ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophils is broken down in the bundle sheath cells to release CO2. This results in increasing the intracellular concentration of CO2. In turn, this ensures that the Rubisco functions as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperature. Based on the above discussion, can you compare plants showing the C3 and the C4 pathway? Use the table format given and fill in the information. Factors affecting photosynthesis, 13.10. Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal and external. The plant factors include the number, size, age, and orientation of leaves, mesophyll cells and chloroplasts, internal CO2 concentration, and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration, water. As a plant photosynthesizes, all these factors will simultaneously affect its rate. Hence, though several factors interact and simultaneously affect photosynthesis or CO2 fixation rate, usually one factor is the major cause or the one that limits the rate. Hence, at any point, the rate will be determined by the factors available at suboptimal level. When several factors affect any biochemical process, Blackman's law of limiting factors comes into effect. This states the following. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of a green leaf and optimal light and CO2 conditions, the plants may not photosynthesize if the temperature is very low. This leaf, if given the optimal temperature, will start photosynthesizing. 13.10.1 Light. We need to distinguish between light quality, light intensity, and the duration of exposure to light. While discussing light is a factor that affects photosynthesis, there is a linear relationship between incident light and CO2 fixation rate at low light intensities. At higher light intensities, gradually the rate does not show further increase as other factors become limiting. What is interesting to note is that light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or in dense forests, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. 13.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increasing concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the levels can become damaging over a long period. The C3 and C4 plant respond differently to CO2 concentration. At low light condition, neither groups respond to high CO2 condition. At high light intensity, both C3 and C4 plants show increase in the rates of photosynthesis. What is important to note is that the C4 plants show saturation at about 360 microliter per liter, while C3 responds to increased CO2 concentration and saturation is seen only beyond 450 microliter per liter. Thus, current availability of CO2 levels is limiting to the C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing the increased rate of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. 13.10.3 Temperature. The dark reactions being enzymatic are temperature controlled. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. The C4 plants respond to higher temperature and shows higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to temperate climates. 13.10.4 Water. Even though water is one of the reactants in the light reaction, the effect of water as a factor is more through its effects on the plant rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by leaves through stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are sites for the light reaction. While the chemosynthetic pathway occurs in the stoma, photosynthesis has two stages: the light reaction and the carbon fixing reactions. In the light reaction, the light energy is absorbed by the pigment present in the antenna and funneled to special chlorophyll A molecule called reaction center, chlorophyll. There are two photosystems, PS1 and PS2. PS1 has 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has a P680 reaction center that absorbs red light at 680 nanometer. After absorbing light, electrons are excited and transferred through photosystem 2 and photosystem 1 and final to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to movement through the F0 particle of the ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecules is associated with PS2, resulting in the release of oxygen, protons and transfer of electrons to PS2. In the carbon fixation cycle, CO2 is added to the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3-carbon phosphoglyceric acid. This is then converted to sugar by the Calvin cycle and the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plant. photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheath cells, the Calvin pathway is carried out for the synthesis of carbohydrates. Thank you.